Welcome to today's lecture. Today we are going to talk about the complement system. Last section we have talked about the activation of classical pathway. And there are two other pathways to activate complement system. The second pathway is alternative pathway. Alternative pathway is independent of antibody. Alternative pathway is triggered by the spontaneously hydrolysis of C3 molecule and produce the C3b molecule combined with a soluble factor B in the serum, and they form the C3b molecule. Then factor B can be cleaved by factor D, and they produce different fragments. The small fragment of BA will be released into the body fluid, and the left C3b-BB is work as a C3 convertase. But in the normal state, C3b and C3bb is not stable. They can be inactivated by the regulatory proteins, such as the factor I and factor H. So in the normal state, there was no activation of alternative pathway. But in the pathogen infection, for example, bacterial infection, C3b and C3bb combined with the bacteria. That means the bacteria provide a platform for their binding. After they bind with the bacteria, they will become very stable. They will not be regulated by the regulatory proteins. And C3b can generate more C3 convertase. And then C3 convertase can cleave C3 molecule into C3b molecules. Then they will form a positive feedback. More C3b and C3bb will be produced. A part of the C3b molecule will then bind to C3 convertase to form the C3b-BB3b, that is C5 convertase in the alternative pathway. Once C5 convertase was generated, then this convertase can cleave C5 molecule to produce the C5b molecules. Then finally, they will form the memory attack complex, leading to the lysis of the bacteria. The next pathway is lactin pathway. Lactin pathway is also independent of antibody. It starts with the mannose binding lactin, that is MBL. We can see the picture. MBL structure is similar to C1q structure with a stalk and 6 heads. And there are also another serine proteases, which is called MBL associated serine proteases 1 and 2. These two proteases are similar to C1R and C1S. So the whole process of the lactin pathway is similar to that of the classical pathway. Let's see how the lactin pathway is activated. This is a bacteria, for example. Bacteria can express mannose, and those mannose can be bound by mannose binding lactin, MBL. After their binding, MASP1 and MASP2 will be activated, and then they will cleave their substances, C4 and C2, to generate different fragments. So small fragment will be released, and big fragment will form a complex, and they exist on the surface of the bacteria. That is C3 convertase, composed of C4b and C2A. When C3 convertase is produced, we can see that is the same as the C3 convertase in the classical pathway. So the following pathway is as the same as the classical pathway. Finally, let's compare these three activation pathways. The alternative pathway and the lectin pathway, both pathways are independent of antibody production. While classical pathway is dependent of the production of the antibody. That means alternative pathway and lectin pathway, they can take effect in the early phase of the infection. While classical pathway will take effect a little bit late. They must take effect after antibody produced. And these three pathways are different in the initial stage. In alternative pathway is activated by spontaneously hydrolysis of C3 molecules. And lectin pathway is activated by MBL binding to the mannose on the bacteria. And classical pathway is triggered by antigen-antibody complex binding to C1 molecule. And classical pathway and lectin pathway, they share the same C3 convertase and C5 convertase, which is composed of C4b, 2a, that is C3 convertase, and C4b, 2a, C3b is C5 convertase in both pathways, which is different from that in the alternative pathway. In alternative pathway, C3 convertase is composed of C3b, bb, and C5 convertase is C3b, bb, 3b. That's difference. But these three pathways are in common in the terminal stage. Once C5 convertase are generated, they will cleave C5 molecule. Finally, they will form the memory attack complex. After the memory attack complex are formed, they will lead the lysis of the bacteria. That is the activation of the complement system. That's all for today. Thank you for your attention.